Who will go first? Hello. Am I audible?
Yes, ma'am.
Yes, ma'am.
So, who is ready to start? Hello.
Hello, ma'am. Hello, ma'am. This is Tejas.
Yes, please.
If you allow us, we can start, ma'am.
Okay. You can start then.
And please enable the screen share.
Yeah, it's there. You can share.
Ma'am, shall we start or should we wait for Anil sir?
No. Meanwhile, he'll join as he finishes his meeting, because right now he is in meeting. But we can start. We'll not wait for him. I'll send the recording of everyone's present to him for his reference.
Okay, ma'am. Hello, Punarva. You can start now.
Yes, sir. Yes, yours, sir. Good morning to all. I'm Punarva, and the topic which I have chosen for the today's presentation is mechanism of COVID-19. And my group members are Akshita KT, Nidhi R, Pratiksha, Priyanka JV, myself, and Tejas Prabhakar and Hemant Kumar. Before going to the actual mechanism of coronavirus and COVID-19, let me give a brief introduction about the coronaviruses. The coronavirus infection has attained a global proportion and has qualified itself to be labeled as a pandemic. The COVID-19 disease is first detected in Wuhan and the Hubei province of China, where a cluster of patients were admitted to the hospital with the initial diagnosis of pneumonia of an unknown etiology. These patients were epidemiologically linked to the seafood and a wet animal wholesale market of Wuhan city. The COVID-19 is a positive strand RNA virus belong to the genus beta coronavirus with the clone-like appearance. This is due to the presence of a spike glycoprotein on the envelope. Because of a clone appearance, it has got the name coronavirus. And one of the important thing in this is we have one of the misconceptions regarding this coronavirus. That is, we always often say that coronavirus isn't new to us, and this word is not actually new to us. We have heard about this next to this rhinoviruses, where coronavirus and the rhinovirus were both caused the common cold. But and also like SARS-CoV-1 during 2003, these are types of coronaviruses families. But the actual particular strain or the SARS-CoV-2, this is a new to us. This is a one which has not been previously identified in the humans, so that we have to come out of this misconception. And another thing which might hit your mind is that what may be the difference between the known strains and this new strain. The known strains which infects the upper respiratory tract, and this is a new strain which has the ability and the capability to enter the lower respiratory tract and reach the lung and cause the pneumonia. And this is a coronavirus which is a zootonic, means the disease spread between the animals and also the humans. And we know that apart from this SARS-CoV coronavirus, the severe acute respiratory syndrome and coronavirus, we have other 6 types which causes the respiratory diseases among the humans. That is human coronavirus 229E, human coronavirus OC43, SARS-CoV, human coronavirus NL63, human coronavirus HKU1, and MERS coronaviruses. And this coronavirus is spherical in shape with 120 to 160 nanometer in diameter, with an outer envelope bearing a 20 nanometer long globe-shaped projections, which collectively resembles like a crown. And we all of us know that on March 11th, the World Health Organization declared this disease as a global pandemic and after that, we are aware that this disease made us to get into the global pandemic situation. Thank you. Now I'd like to hand over this to Pratiksha.
Good morning, everyone. Now we will see the classification of coronavirus. The virus, mainly the viruses can be classified according to the morphology. That means the structure of protein and the structure of virus. For example, the corona, which is identified in China, is actually different from the coronavirus, which is identified in India. And mode of replication and the host organism, like the viruses can infect animals, bacteriophages, and viruses can infect plants. But the coronavirus is a animal virus which can infect both mammals as well as humans. And the nucleic acid type, like double-stranded RNA virus, single-stranded RNA virus, double-stranded DNA virus, and a single-stranded DNA virus. But the coronavirus is positively single-stranded RNA virus. That means RNA viruses usually usually do their transcription in the cytoplasm, which do not integrate their genetic material into the host genome. And now let us look at the biological classification. International Committee on the Taxonomy of Viruses, ICTV system, will formally classify the viruses, and the Baltimore classification system is a type of classification of viruses which group viruses into 7 groups based on their manner of mRNA synthesis. And coronavirus is belong to the phylum Psicoviricota and the order Nidovirales and the family Coronaviridae, subclass Orthocoronavirinae, and genus Alpha coronavirus or a Beta coronavirus. Alpha coronavirus can affect both mammals as well as humans, but Beta coronavirus will affect mammals other than the humans. Next, let us look into the mode of transmission. The transmission of coronavirus can be transmitted by droplets or airborne transmission. Droplets are larger particles like salivary particles or respiratory droplets, which can be transmitted from the infected person to the other person by direct or indirect or a close contact. And when a person who coughs or sneezes, the droplets may transfer to the surface, so through which in an uninfected person can be infected. In airborne transmission, it is like aerosol materials which are smaller than the droplets, and they are in the air. When an uninfected person inhale the aerosols, that virus can be transferred to the person, and which causes respiratory disorders. Now I would like to invite Tejas to continue the presentation.
Good morning, everyone. Myself, Tejas Prabhakar, and I'm here to explain about mechanism of action of COVID-19. Let me keep it simple. We are going to start at the end, and we are going to look at 3 things that actually result in death of a person who is infected with COVID-19, and look how those 3 things arise through the actual infection. So the biggest thing that results in the death of someone who is infected with COVID-19 is multi-system organ failure, and that happens because of 3 things. There will be decrease in blood pressure. There will also be decrease in blood volume. There will also be decrease in perfusion. Perfusion is basically the delivery of oxygen to our organs. So ultimately, what's happening here is organs start to fail because they are not having enough oxygen delivered to them. That's what results in organ failure. and that what results in the death of a person who is infected with COVID-19. So let's see how all these three things happen. That is, why does there is a drop in blood pressure, and why does there is a drop in blood volume, and why does the perfusion drop? Let us start with the coronavirus, that is coronavirus. This is the simple diagram of coronavirus. These spikes on the virus are called glycoproteins. They act like a kind of key that can go into a lock. This virus has a genetic material as RNA, that is ribonucleic acid. It's like an instruction manual which contains all the information that the virus needed to make the copies of itself. It's like an instruction book on how to build a virus. A protein shell provides a hard protective enclosure for the genetic material as the virus travel between the people it infects. And the outer envelope of the virus allows the virus to infect cells by merging with the cell's outer membrane. This coronavirus use their spikes like a key to get inside a cell in your body, where it takes over its internal machinery, which makes the host cell to build viral components. When an infected person talks or coughs or sneezes, droplets carrying this virus may land in our mouth and nose, and then it goes into our trachea, then to bronchus, and then to bronchioles. It goes right to the end of the tube, so where we got alveoli. Yeah, here is this simple diagram of alveoli on your left side. Alveoli are the very important place because it's the place where our oxygen can transfer into our bloodstream to oxygenate the blood and get circulated around our body. When we breathe in oxygen, oxygen moves into the bloodstream from the alveoli, and when we exhale, the carbon dioxide moves out from the alveoli. The image on your right side is the magnified image of alveoli. The cells we are really interested are these bigger cells, which are 3 in number. They are known as type 2 pneumocytes, and their main function in the lungs is to produce the surfactant inside the alveoli. This surfactant is produced to form a kind of coat inside the alveoli to look that alveoli are not sticking together, because alveoli is going to be damp in there because the air is kind of moist, and this surfactant stops alveoli sticking together when we breathe in and out. That is, the surfactant helps in elasticity and the stretchiness of our alveoli, which makes us easy to breathe. So these pneumocytes produce that surfactant. But on the outer side, as you all can see, the image on your left side is the magnified image of type 2 pneumocyte. On the outer side of the pneumocyte, we have got the orange kind of blob, which is called the ACE2 receptor or ACE enzyme, which stands for angiotensin converting enzyme. This enzyme converts something called angiotensin 1 to angiotensin 2. Angiotensin is produced, angiotensin 1 is produced when you have a kind of low blood volume and a low blood pressure, and this enzyme turns angiotensin 1 to angiotensin 2, which raises the blood pressure. This converting enzyme is generally where the virus ends up entering the cell. The glycoproteins on the virus, which are like little keys, interact with the ACE enzyme here, and that results in the entry process of virus, which is called endocytosis. This is how virus takes over cell's infrastructure inside to basically turn this type 2 pneumocyte into a virus producing cell. When the virus comes in, as you all can see on the image on your right side, when the virus comes inside the cell, it unwraps itself. The little strand of RNA, which is the instruction manual, is released alone. As you all know, every cell consists of ribosome, which is basically a protein producing factory. It reads the instruction on the RNA, and the protein is made out of it. So as usual, it also reads this viral RNA, and the proteins are made out of it. One of those proteins is RDRP, which stands for RNA dependent RNA polymerase. Now this is basically a photocopier for the viral RNA. So this instruction manual basically go through this RDRP, and this makes the multiple copies of the viral RNA, and eventually all the viral proteins overtake the cell, and cell become the viral factory, and the viruses start to build up inside the cell. So we have got two things going on here. We have got a ribosome making all the proteins and little bits and pieces needed to build a virus, and also a little photocopier for the RNA. So eventually the viruses are released out of the cell. So now let's go to alveoli. As you all can see here, type 2 pneumocytes has already released viruses into the alveoli. Once there is any infection, our immune system kicks in, and the first thing that is going to appear are neutrophils, as you all can see here. There's also macrophages present here. This effect of our immune system releases some chemicals such as chemokines or interleukins. These are little chemicals that help to coordinate the process of our immune system. These are like the signals that attract more cells to find where the problem is. Some of those chemicals which are released are interleukin 1, interleukin 6, and TNF alpha. TNF alpha stands for tumor necrosis factor. This is actually a pyrogen. It helps in elevating the body's temperature. Since this TNF are released into bloodstream, they end up going to the part of the brain called hypothalamus. They have an effect on the hypothalamus, and hence there will be increase in our body temperature, and that is why the person who is infected with COVID-19 experiences a fever. And at the same time, in and around our alveoli, there will be an increase in the permeability of these cells. Now what happens is the liquid part of your blood start to leak into the area between your alveoli and inside the alveoli. So there will be a buildup of fluid inside the alveoli and around the alveoli, which is very dangerous because remember I said that the alveoli is the place from where the oxygen goes into bloodstream. But that doesn't happen now because we have got this fluid filled building up fluid filled inside the alveoli, and within the alveoli space, this condition is called alveolar edema. Ultimately, what happens is our oxygen levels falls because we are not getting the oxygen into our blood, and also the surfactant will not be produced by pneumocytes because there will be fluid filled in. So ultimately, there will be difficulty in breathing. That is why the COVID-19 patients are provided with ventilators to assist them in breathing. Those interleukins and tumor necrosis factor, which are chemicals produced by our immune system, has an effect on our circulatory system also, which results in systemic inflammatory response syndrome. As this condition develops in initial stages, you have got only few cells that are infected. But as the virus spreads through the cells of respiratory system and are starting to become infected, our immune response also goes up and it gets stronger and stronger, and more of these chemicals are released that has wider effect all over the whole body. There will also be increase in the permeability of cells around our circulatory system. So eventually what happens is the fluid in our blood stream leak out all over the circulatory system because the blood cells start becoming leaky. Remember, blood cells start becoming leaky. So remember I said what results in death of a person who is infected with COVID-19 is there will be drop in the blood volume. This is the reason why there will be decrease in blood volume. That is, the liquid part of your blood starts to move out of your bloodstream into your tissues. Since the blood volume is decreased, it also results in low blood pressure, and there will be no proper supply of oxygen. Since alveoli is filled with fluid, there will be a drop in perfusion. That is, there will be decreased delivery of oxygen to the organs. Ultimately, all these three things result in multi-system organ failure, which causes the death of the person infected with COVID-19. I think I have made a sincere attempt. Now I'll pass on to my teammate Hemant Kumar. Hemant, over to you.
Thank you, Tejas. I'm going to explain the day-to-day symptoms. Day 1 to day 2, the beginning symptoms are similar to the common cold with mild sore throat and neither having a fever nor feeling tired. The patients can still consume food and drink as usual. Day 3, the throat starts to feel a bit painful. Now, body temperature reads at around 36.5 degrees Celsius. Although it's uncommon, other symptoms like mild nausea, vomiting or wild diarrhea are possible to set in. Day 4, the throat pain becomes more severe. Other symptoms like feeling weak and joint pain starts to manifest. The patients may show the temperature reading between 36.5 to 37 degrees Celsius. Day 5 to 6, the mild fever starts. Then the temperature reading will be above 37.2 degrees Celsius. The second most common symptom, the dry cough, also appears. Dyspnea or breathing difficulty may occur occasionally. The most patients in this stage are easily feeling tired. The other symptoms remain about the same. These 4 symptoms are among the top 5 key indications of COVID-19. Day 7, the patients that haven't started recovering by day 7 get a more serious cough and breathing difficulty. Fever can get higher up to 38 degrees Celsius. It can lead to develop a further headache and body pain or worsening the area, if there any. Day 8, the symptoms are likely to be worsened. The severe shortness of breath become more frequent. The temperature reading goes well above 38 degrees Celsius. At this stage, without a medical consult, may lead to a serious issue like respiratory distress and also loss of appetite, and may be facing abdominal pain. And for the minority patients, the fragile group must prepare for the possibility of acute cardiac injury or kidney injury. The vulnerable patient may develop a secondary infection caused by the new pathogen in the lower respiratory tract. This severe condition may then lead to the blood coagulation and ischemia, and finally death. Now, the prevention measures to control this COVID-19 will be explained by Priyanka. Over to you, Priyanka.
A very good afternoon to everyone. Myself, I'm Priyanka. I would like to talk about preventive measures of COVID-19. Preventive measures are always prepared by knowing how a virus transmission occurs. Since the coronavirus transmission is only by contact, droplet, and airborne methods, so we should create a kind of barrier for these 3 to avoid the virus to reach the sensitive part of our body through which the virus can enter the human system. So first of all, isolation of confirmed or suspected cases with mild symptoms is recommended. So this is what we call it as quarantine, because isolating them would be easy for others to not get infected. And wear a mask when physical distancing is not possible. Physical distancing is about 5 to 10 meters for from one person to another person. So if it's not possible, we should wear a mask to keep our sensitive parts, that is nose and a mouth, to avoid the contact of virus. So next, maintaining a safe distance from anyone who's coughing or sneezing, because they contaminate the surrounding area through through coughing or sneezing, by emitting out the viruses through the air to the environment. So we should maintain a minimum distance from the person who's coughing or sneezing, and clean your hands often. When you're outside, use sanitizers, and when you're inside, wash your hands with hand washes, and do not touch your eyes, nose, and mouth too often, because that is the only way through which virus can enter our body. And cover your nose or mouth with your bent elbow or a tissue when you cough or sneeze. It's not only that you should avoid you getting in contact with the virus, but it is also that you shouldn't contaminate the surroundings, because it could be a dangerous thing for another person who's next to you. So that is why always bend your elbow, so that you wouldn't be transmission project or something like that. So in addition to this, through our presentation, we would like to add an additional information regarding the assignments given to us. Since it's about the antiviral property of milk and mechanism of COVID-19 or coronavirus, we can combine these 2 ideas actually. So some articles and journals talk about antiviral action of milk against human immunodeficiency virus, which is known as HIV, and the human cytomegalovirus, which is known as HCMV, which are human affecting viruses. But note this point, that unmodified milk milk proteins are generally not active against these viruses. So if we are using milk as an to get the antiviral action, then definitely we should modify the proteins or the molecules which have the antiviral property chemically. So if we modify them chemically, then it would be of good use. So an exception is that lactoferrin, which shows a significant antiviral activity against both HIV and HCMS. This could be another way or one of the substitute to manufacture the vaccine, or simply to tell this as a remedy to avoid COVID-19, since it is also an human affecting virus. Thank you, one and all. That's it.
Thank you, everyone. That's it, ma'am.
Okay. I would say it's a really impressive presentation. And now, before I give any comments, I would like to request all the participants and all your friends to discuss with you guys if they have any query, or if they feel that this should be there in the presentation, or some information are missing, or they want to incorporate something in that. You can open up for the suggestions and for the discussion. Please.
Yes, ma'am.
If anyone has a suggestion to make, you guys can always give us. And if you guys have any queries or questions to ask us, you're free to call, ask us.
Any argument or discussion for the particular thing? Those who have already presented on COVID-19 can also discuss with these people. See, if you will not actively participate in these presentations and assignment, then it's of no use of doing this, right?
I just want to give one suggestion. You guys could have used tools like BioRender for explaining the mechanism of COVID. So it would be much better. Like, yeah, instead of it was good those hand-drawn figures, but in future maybe you can try tools like BioRender also.
Yes, Akshay, but yes, I accept your suggestion, but I tried to keep it simple. Yeah, of course, we'll use. Thank you for the suggestion.
Hello.
Yes, please.
Good morning, ma'am. I'm adding some apart from the COVID-19 mechanism. Spike is the main protein that interact with the host cell receptors in the first step, as of receptors binding spike protein, as you all know. So spike protein is cleaved into S1 and S2 by the host cell protease, one of which is a transmembrane protease, serine 2 abbreviated as TMPRSS2. Then the main function of S1 subunit is to bind with the host cell surface receptors, while the function of S2 subunit is to mediated membrane fusion. So one of the potential therapeutic approaches is to either develop vaccine that contains antigens derived from the spike protein, which can boost recognition of the virus by the immune cells, or to develop monoclonal antibodies that bind to the coronavirus spike protein and block the interaction with human cells. Thank you.
Thank you, Devraj, for the information. Ma'am, shall I stop sharing the screen?
Yeah, sure. Okay, So I have few suggestions on the making of presentation. You have not included the references, which is most important part. So whenever you are sending me this presentation, or if you have sent, please resend me with the proper referencing from where you have took this information, for whether it is a prevention or mechanism or whatever it may be, right?
Okay,
It's
ma'am.
very
Sure.
important. Without referencing, it is not accepted, right? And what else was that? Yeah. Do you know how to cite the reference? If you don't, then we'll keep one session for this, how to do citation and how to write full length references before your review writing assignment. Okay. So for citing this
Yes,
presentation,
ma'am.
okay. So if you don't know the citation in the presentation, please let me know. I'll send the template for that. Otherwise, if you know, then you can include the referencing in your presentation and send me back. And apart from that, your presentation was excellent. But I'll say that there are some slides you can incorporate few explanation, so that it could be it can be a self-explanatory, right? By looking at your slides, if I'll send your presentation to any reviewer, he or she may be not going to understand, as specifically if I'll talk about the symptoms which explained by someone, I don't know, from your group, who you have just listed out fever, then cough and chest pain and whatever, and then you have explained their limitation range of temperature in the fever, and whatever may be characteristics of each and every point, right?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
I think you have presented it very well. It is very clear, but I need to, as a presentation, as your assignment, I need to send it to the other reviewer. He will not going to understand what you have presented, right?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
So I suggest, if you are actually orally presenting, that way your presentation is perfect. But as an assignment, you need to incorporate each and every, at least not in detail, but you should mention those important information in the slide.
Okay, ma'am. Sure, ma'am.
Okay, ma'am. Okay, ma'am.
And
Okay,
please,
ma'am.
content slide. I don't remember whether it was there or not. The content slide, what you are going to cover in your presentation.
Okay, ma'am.
Okay, you should keep that, write all the members name that you have mentioned, I think, and then please incorporate all the important information in each slide, and cite the proper reference from where you took this information.
Okay,
And
ma'am.
at the end, you
Okay,
can list out the detailed referencing in the reference slides. And one more thing, if you have some conclusion, or you have already referred so many things to make this presentation, so I suggest you can keep the conclusion slide also,
Okay, ma'am.
if it is possible. Otherwise, that's fine.
Okay, ma'am. Sure.
Yes,
So
ma'am.
please note down these comments and improve your presentation and send it to me.
Okay, ma'am. We'll send
Okay,
it.
ma'am.
Right? Okay. So it was very nice and excellent.
Thank you so much, ma'am.
Thank you, ma'am.
thank you, ma'am.
Thank you. Thank you so much.
Thank you, ma'am.
Okay. Who is next?
Hello, ma'am.
Yes.
Hello, ma'am. This is Soham. We are ready with our presentation, ma'am.
Yes, please. Please.
Okay. Nitish, can you share the screen?
Ma'am,
Yes.
can I share the screen, ma'am?
Yes, yes, please.
Yes, ma'am.
make it full screen. Okay, thank you. So, ma'am, should we start?
Yes.
Okay, so good afternoon to all the respected dignitaries and the participants of BIIS-7. Today, I, Soham Datta, along with my group, is going to present before you a presentation on antiviral properties of milk. First, let me introduce you with my fellow group members. First is Janak Dave, who is a third year student of pursuing BE Biomedical Engineering at Sri Ramachandra Engineering College, Coimbatore. Secondly, myself, Soham Datta, I am pursuing the B Farm at second year from Dr. B.C. Roy College of Pharmacy and Allied Health Science. Next, Nitish Kumar R, fourth year student, B Farm, JSS College of Pharmacy, Uti. Next, Bhavni Krishnan, who is a third year student of BSc Biotechnology, studying at Nehru Arts and Science College, Coimbatore. Next is Prachi Sharma, fourth year student, BTech Biotechnology, Bansal Institute of Engineering and Technology, Lucknow. Next is Sweta, a third year student of BSc Biotechnology, MES College. So the next slide, please. Okay, so let me take you with the table of contents, which includes introduction, the non-proteinaceous antiviral parts in milk, mechanism of action of antiviral substance, mechanism of viral inhibition of lactoferrin, effect of immunoglobulins from colostrum, and study on various nutritional components of camel and human milk, and lastly, the conclusion. Next. So let me start with the introduction. So milk, as we all know, is thought to be the main source of bioactive molecule, especially for infants, providing antibacterial and antiviral properties, facilitating the nutrient absorption, promotion of bone growth, enhancing the immunological protection, and supporting the host immune competence. Secondly, the antiviral activity through the immune stimulation and suppression of the host immune inflammation are due to the presence of casein proteins, whey proteins, and their derived peptides in milk. It is found primarily from in vitro studies that certain milk proteins interfere with infectious agents. Casein proteins mainly function as antiviral by regulating the innate immune response through upregulation to enhance the killing of virus. The whey protein, lactoferrin, and pepsin derived peptide fragments of the protein, that is lactoferricin, is also very important due to its direct interaction with viral particles and cellular receptors on target cells. Studies have demonstrated that active milk protein and peptide compounds can be extracted from a variety of species, including humans, bovine, porcine, mice, and camel. Proteins except lactohydrin and peptides such as glycomacropeptides put together interfere with the infection for some virus. Chemical modifications of milk proteins that cause changes on proteins and responsible distribution enhance their effects against certain virus. Next slide. Now, here are some antimicrobial properties of lactoferrin. It acts as an immunomodulator, as an antifungal agent, as an antiparasitic agent, antiviral, antibacterial, protease inhibitor, ribonuclease, procoagulant, promicrobial, auto antibodies. It helps in granulopoiesis, transcription factors, etc. Next slide. Now, lactoferrin has a broad spectrum antiviral effect. It has the antiviral activity on human rotavirus, human echovirus, human papillomavirus, hantavirus, herpes simplex virus type 1 and 2, influenza virus, poliovirus, hepatitis B and C, adenovirus, and human immunodeficiency virus. Now, the benefits of milk, which we all know, is for maintaining healthy teeth, healthy bone, enhancing the hydration, glowing skin, lowering the risks of cancer, and it also aids in weight loss. Now, it's up to you, Nitesh.
Thank you, Son. It's my part to store its non-proteinase parts of milk. Apart from protein, there are many components in the milk has been acting as an antiviral agents. According to the Wells, Alkanes and Collins report, the lipids of the milk as exhibiting the antiviral property against Semelic sporus virus and herpes simplex virus 1 and CMV. Later study by Isaac Thomas and Pesolano 1986, he verified that antiviral properties of some of the fatty acid present in the human milk also. When it comes to fatty acid has the main impact on the enveloped virus like herpes and rubus viruses, but was not effective as other on non-enveloped virus like polio viruses and vaccinal viruses. It the mechanism of action of the antiviral property of the fatty acid comes to the pot of bilayer pot of envelope. It describes the bilayer pot of envelopes and it drains out the entire particles which present inside the pathogens or viruses. According to the report of Isaac, the medium chain saturated and long chain saturated fatty acids was very less competitive against the envelope viruses than the short and long chain saturated fatty acids. From the report of Thomas review, the monoglycerides of the active fatty acid has higher activity than the free fatty acid which are present in the human milk. Okay. Let me come to the mechanism of action. The mechanism of action has been classified under 4 categories. By inhibiting the entry of viruses, inhibiting the viral interaction with the viral capsid and pathogenic, that is the structural change in the host cells, and inhibiting the replication of viruses. Let me come to the mechanism of action of lactoferrin. The lactoferrin prevents the virus attachments to the cells, which is represented in the picture which is present on left side of the slide, and it also inhibits the virus replication by inhibition of interferon alpha and beta. When it comes to inhibiting the entry of viruses, the main reason behind it is electrostatic interaction. Due to the electrostatic interaction change in the surface of the host cells, the virus entry has been inhibited in many cases. When it comes to the interaction with the viral capsid, the main change in the alteration in the viral capsid also causes the or prevents the entry of viruses into the. When it comes to the structural change in the host cells, the lactoferrin gas interact with the glycoaminoglycogen, which is present on the surface of the glycogen, which is the key or main part of the host cells to get interact with the viruses, which helps in the glycoaminoglycans, which help in the attachment, proliferation, migration of the viruses into the host cells. Hence, it get interface with the host cell to develop into it. The lactoferrin inhibits these glycoaminoglycans. Due to this, the entry of viruses has been prevented to the core. The inhibition of replication of viruses is okay. When it comes to the inhibition of virus replication, the RNase plays a major role in it. It actually iterates the RNA at RNA DNA hybrid. The picture shows that from the picture, the RNase plays a role in iterating the RNA part in RNA DNA hybrid. Due to that, there is a cleavage on the cyclopeptide bond which takes place between the DNA. Due to that, the replication has been inhibited, and the reproduction of the virus is also inhibited. These are some examples for the antiviral activity of lactoferrin against the viruses. When it comes to herpes viruses, the lactoferrin has inhibiting the virus development by binding to the both virus particle and the cellular particle to prevent the absorption and entry, interfere with the intercellular replication event or synthesis of the progeny virus indicator. When it comes to non-enveloped viruses, the poliovirus is lactoferrin, which inhibits the proliferation of the lactoferrin by binding to the viral receptor on the target, especially glycoaminoglycogen, which binds to it and alters it, the structure of host cells, which prevents the entry of viruses into the host cell. Janak, you can continue.
Yeah, so good afternoon all. So we all know that in milk there are, they say that it has immunoglobulins, but if you see, not all milk has immunoglobulins. The concentration would definitely vary. Now, if you look at the colostrum, now colostrum is something again present in the milk that is yellow in color, and you can find it like when a human or a cow has given a birth, the color of the milk is not going to be white directly, it's going to be yellow in the starting. Yellow is because presence of colostrum. Now what is colostrum? So the yellow coloration of colostrum is because of beta carotenoid, is also present in few plants. Yes, and in humans, what colostrum does or how it affects or helps us in antiviral thing is, colostrum has a lot of immunoglobulins. Its concentration is way high. You cannot actually just take immunoglobulins, just say like, what if I take my vaccine which has immunoglobulins and I just take a sip of it, because it's going to get digested, right? So the concentration in immunoglobulins of in colostrum is way high, and colostrum is also quite sticky. So when, as soon as it reaches the cell lines or in stomach, they tend to stick to the linings also. Now what happens is, when they stick, immunoglobulins try to diffuse inside the cell line itself and gets carried in the blood. And immunoglobulins actually get absorbed in the duodenum jejunum, the curve of the stomach, right? The very starting, it gets absorbed there itself. So it does not get degenerated into your intestine and all. It's effectively taken up in your blood. So how colostrum also helps a lot is like, as I said, immune support system. It protects your gastrointestinal tract. It has antibody levels 100 times higher than the regular cow milk. So if you take the fresh milk, this very fresh milk of a cow, it would have a higher immunoglobulins. Then about gut health and microbiome. So colostrum not just is antiviral activity, but is also sometimes probiotic. It helps in the growth of few useful bacteria that is necessary for our gut integrity. Also about skin health, what happens is, in colostrum there is a little amount of telomerase which helps in cell regeneration, but you don't find this cell regeneration activity in milk that we drink, because we denaturate, we boil the milk a lot, and the telomerase gets denaturated and it's not very effective. But if you drink the raw milk, it could be. And also it contains lactoferrin. I think my friends definitely said how much lactoferrin is important. That also helps in maintaining skin cells. Next slide.
Hello
Prachi.
everyone, I'm Prachi Sharma and I'm going to tell about different milks. First is camel milk. It contains the protecting protein like neuraminidase, lactoferrin, lactoperoxidase, NAGase, PGRP, immune gamma globulin, and immunoglobulins. They are naturally present in the body and act as an antimicrobial and provide immunity. It naturally has the antiviral, antifungal, antiparasitic, and growth promotion activity and anti-tumor activity. lysosomes. Camel milk has lysosome, also known as muramides. It is also an antimicrobial enzyme produced by animals and form a part of innate immunity system. It acts against inflammation, cancer, viruses. Immunoglobulins, it gives immune protection. As we know, these are antibodies produced by plasma cell or white blood cells. They act as critical part of immune response by specifically recognizing and binding to a particular antigen. Next is lactoferrin. These are iron-binding protein present in large quantities in milk. The main function is non-immune protection. It is active against both DNA and RNA viruses. The antiviral effect of lactoferrin lies in the early phase of infection. Ariodactyl milk contains different peptides and protein which act as different role in absorption, digestion, growth, and immunity. As we already said, it participates in different functions of the body. Next slide, please. Next is human milk. It provides the protection for baby against infectious disease. Human milk contains 0.8% to 0.9% protein, 4.5% fat, 7.1% carbohydrate, and 0.2% minerals. Carbohydrates are mainly lactose. This protection is presented through many mechanisms. First is specific protein targeted protection against pathogen. What happens is the immune system responds to antigen by producing cells that directly attack to pathogen or by producing special protein called antibodies. Antibodies attach to an antigen and attract cell that will engulf and destroy the pathogen. Next is broad spectrum. Broad spectrum mechanism works against a wide range of disease causing microbes. Next is non-specific protection. It includes physical and chemical barriers, the inflammatory response, and interference. These are germicidal effect, organic process action, lysis of microorganisms, antiviral effect, antiprotozoal activity, and substance action. Additional to dose protective function of the protein and lipid of human milk, many enzyme gives protection by generating parts that are germicidal, stop inflammatory reaction, or depend the integrity of protein. Next slide, please. And the conclusion is, if we do chemical modification of milk protein that cause changes in charge of protein and accountable distribution enhance their effect against bound viruses. The medium chain saturated and long chain unsaturated fatty acids were less complicated against self-enclosed viruses than short and long chain saturated fatty acids. Lactoferrin acts as binding site for several viruses on the cell surface in the mechanism of microorganism inhibition by lactoferrin. As we already mentioned, artiodactyl milk is used to treat sickness in jaundice, malaria, TB, and also for treatment of redness, asthmatics, abdomen discomfort, HIV, hepatitis, and urinary problem. It also contains peptides and protein that exhibit biological activity useful in several bioprocesses. Human milk protect the infant against infectious disease, specific target protein, and non-specific broad-spectrum protein protection is provided by human milk through distinct mechanisms like germicidal effect, antiviral effect, microorganism lysis, antiprotozoal activity, etc. Thank you so much.
Hello.
Yes.
Okay, I am adding the other conclusion apart from the antiviral properties of milk because I have studied one international dairy journal, 16 2006, antiviral properties of milk, proteins and peptides. I studied their review. So as per their review, that I'm telling the conclusion. So the summaries that result of research undertaken to date primarily under in vitro condition. In more recent years, in vivo effects have been reported in mouse and rat models, but there is still a need for further studies aimed at elucidating the mechanism of antiviral action of milk protein in vivo. In the immediate future, milk proteins and peptides, with the possible exception of products containing immunoglobulins, are not likely to be used in commercial application in prevention and therapy of viral infection in animals and humans. Further research is needed to provide substantive evidence for the potential in vivo effects that could be attributed to native and modified milk proteins and peptides derived from them. Once this is unequivocally established, the benefit of some of the chemical modifications observed in vitro could be explored for optimization of preparations for a specific application in animal and human healthcare. Thank you.
Hello.
Yes.
I would like to say to the team who presented right now, there is no need to read the slides. Okay. Don't put too much in writing in your slides, and there is no need to just read it, go through it. You just put your points, important points in in bullets, you can add it, and then you explain what that points mean. So the presentation should be in this form. So most of the slides was like you all were reading it. So don't do it. And also from wherever, as Megha has already, you have told earlier also that from wherever you are taking the pictures or images, cite it, give the references. It is very, very important. Is it clear?
Okay, ma'am. Okay, ma'am. Thank you.
And don't read the slides, please. Add the important points in bullets, and then you can explain it. Clear?
Okay, ma'am.
Yeah, so the presentation was again good. You have put your efforts, but I'll say that I agree with Dr. Shaila that you should not read while presenting. But I think this is the assignment presentation, so whatever you have incorporated, that is fine. But when you are presenting, you should not read as it is written, right?
Okay, ma'am.
Yeah. So another thing is, again, the citation, you have the same comments for as earlier group have, that you should cite the reference from where you have taken the information, and then at the end you can give the full length references in the reference part. And another thing is, I told to compare the antiviral activity of 3 milks by keeping the table. That would be more explanatory, right? You can compare that whether camel milk, human milk, or what other milk you have taken. You can compare that these are the composition differences, and these are the uses of 3 milks to fight against the viruses, different viruses, whether it is, and see, the Professor Gupta was more, he gave the example of that farmer who are, sanitizing their hand with milk before sowing the seeds, right? Or maybe dipping the seeds in the different milk, goat milk or camel milk. So he was talking about the viruses which are infecting the agriculture field, right? And you have not included agriculture pathogens. I mean, viruses which are highly affecting the agriculture field. You should include that. Is that clear?
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Yes. So you can do that. You can incorporate all these comments into your presentation, and then you can send it to me.
Yes, ma'am.
Okay, ma'am. Absolutely.
Okay. So and other students who wants to discuss with these group, any comment or any argument, suggestions, anything, if you want to do with these students. So the main motive behind the keeping the presentation is to build your confidence, and you will get interest, and other students can also benefit it from this. So that's why I'm continuously asking you to discuss the things, or you can argument, or you can do you can give suggestions for any topic, right? So please be interactive or actively participate in this.
Excuse me, ma'am.
Yes.
Ma'am, can we have later words session for the citations and all? Because we want to learn it. Many of the participants don't know proper citation.
Sure, that I'll keep, because our session on research methodology has been postponed. So I'm trying to get a proper person for this session. So
Sure, ma'am.
that I'll do. Anything else?
No, ma'am.
Okay. So
Excuse me, ma'am.
please.
Ma'am, when we are going to have our next presentation, like we hadn't given our presentation right till now.
Yeah, so we are continuously doing that. We have continued. This session is continue now. You can present it.
Ma'am, right now all my team members are not present actually.
So that's why I am already pre-informed that today we have presentation session. So you should have told them or informed them to be present here. So who is next to present?
Ma'am, can I, ma'am?
Yes, please.
Okay, ma'am. Thank you, ma'am.
Excuse me, ma'am.
Mm-hmm.
Can we submit our presentation tomorrow, if possible?
Yeah, no problem, no problem. You just improve your presentation, both the team and earlier team also. Please improvise your presentation and then resubmit to me.
Okay.
Can I continue, ma'am?
Yes.
Is the slides visible, ma'am? No?
Yeah, you can keep a slideshow.
Okay, ma'am. Okay, my name is Devra Saraswati, and I'm going to my presentation on my topic is the drugs-induced ototoxicity during pregnancy and risk factors for congenital deaf and mute. I think this topic can help all of you because what is the deaf and mute, and what is the disorder of your hearing, and what is the ototoxicity. So a lot of people has not understood apart from this. So I think it can help for as a awareness purpose for deafness, prevention of the deafness, and congenital also congenital deafness. And also which drugs used on the ear that to show the ototoxicity in the ear. So I will discuss apart from the ototoxicity, which drugs induced, and mechanism of the hearing, and part of the ear, which organ of the ear are mainly involved for the hearing mechanism purpose. So first introduction apart from the ototoxicity. So definition is damage to the cochlea. So here mainly in the ototoxicity means whenever you take the ototoxic medication like azithromycin and all of the mycin group medicine. So so mainly your cochlea has damaged due to chemical exposure. Okay. And then mainly in the cochlear vestibular apparatus also exposure, and the drug ototoxicity also is defined as a temporary or permanent inner ear. Okay, so it can be ototoxicity can temporary also permanent inner dysfunction after drug exposure, so resulting in the hearing and balance disturbances also. So we all know our ear part is mainly functioning the hearing. So this is not also we both ear know has functioning the balancing of the body. Okay, so our whole body, suppose you have observed in some other peoples that have any hearing loss and difficulty of the hearing, then they people disturbing the balancing of the body, right? Like turn hand behind on the ear. So this symptom is also, and you can see in this picture, no. So this picture of this baby. So here no pinna is present in on the skull. Okay, so that means the anosia. So it's called as a anosia. Anosia means the no pinna. So also you all about know about the what is the pinna, because just I will discuss the part of the ear. Okay, and which part and which mainly ear are divided into three parts. So here this picture, that means the here no pinna, that is the anosia. So also so no pinna, that means no, this is the not mandatory for this baby can't hear. So it need to be the detailed evaluation of the ear in the medical as well in the hospitals. Okay, so after that we find that the what is the deformity of the pinna and what is the deformity in the structural anomalies. Okay, so here you can see that this picture of the cochlea. So cochlea means that so here this cochlea, snail shaped cochlea, and then the remaining part, this is the semicircular canal and electrical saccule vestibular, and this picture is the eardrum. Okay, eardrum, and three small bones like malleus, incus, and stapes. So you all, I think, know the whole the smallest bone in the whole body is the stapes. Okay, so this picture of the steps, and steps connected to the oval window of the vestibule. So vestibule, that means this part of the vestibule, and vestibule connected to the cochlea. Okay, so that means I am telling that the part of the middle ear. Okay, so this picture like eardrum to these steps are called as a middle ear. Okay, and then from the semicircular canal and the cochlear vestibular present there is called as a inner ear. So main inner ear part is the semicircular canal, and also yet utricle saccule and vestibule and the cochlea. Okay, so this is the main part of the inner ear, and our hearing functioning. So the mainly our inner ear parts. Okay, and also help to the also helped middle ear as well, external ear. Okay, so just I'm telling that the part of the which part are involved in the middle ear and which part involved in the inner ear, and what is the main part for the hearing also balancing purpose. Okay, so eardrum, malleus, incus, and the steps. Then also you can see the outer ear, middle ear, and the inner ear. Okay, so just you can see that this is the pinna. This whole the like S shape. This is the pinna part, and this is the ear canal, also auditory canal. Okay, and then you can see this blue picture is the eardrum, that is also called as a tympanic membrane. Okay, this is the tympanic membrane, and this is also just I told the malleus, incus, and steps. So you can see that these steps now connected to the oval window. Okay, oval window and dot window. Two window opens on the vestibules. Upper part are the round window, and the lower part is the oval window. But our steps connected to the oval window, that can help the fluid mechanism in the cochlea to the brain. Okay, and then this is the hostage and tube. Okay, to the pharynx, because our ear, nose, and the eyes also interconnected. So that means that these pathways go to the pharynx. So pharynx also is our the oral structure. Okay, so this is the pharynx, and then the Eustachian tube also connected to the middle ear. And the middle ear, I told what is the middle ear, which part involved in the middle ear, just eardrum. So we can't the eardrum part involved in the middle ear, just tympanic membrane, that means the eardrum partially divided external ear and the inner ear. So midline of the tympanic membrane. So this is the showing the midline to the external ear as well the inner ear. So this is the midline. So this is not a part of the external ear and the middle ear. Okay, but middle ear, just I told this small bones also called as the ossicles. Okay, so this is also malleus, incus, and steps are interconnected. Okay, And apart from the eustachian tube, this is the this work of the ventilation. Okay, so ventilations in the middle ear. Okay, suppose you close your nose and like some few after few seconds you open the nose. Okay, so you feel the like some air go through the this way. Okay, like your pinna has some vibration occurs. Okay, so you feel मेरा कहने का मीन्स ये है कि जब हम नोज को बंद करते हैं तो हम ये फील करते हैं कि कुछ एयर पैसेज जो है वो हमारे एक्सटर्नल ईयर से होकर बाहर निकल जाते हैं। ठीक है, ये हमारे क्या होता है, ईयर कैनाल से होकर बाहर आ जाते हैं। तो कहने का मीन्स क्या हुआ कि ये जो हमारा ऑस्टेचियन ट्यूब है ये क्या करता है, वेंटिलेशन का काम करता है। इट वर्क एज ए वेंटिलेशन टू द एक्सटर्नल ईयर। ओके, and also apart from the cochlea, it is the snail shape. Okay, and then the in the main organ of the coti involved in the cochlea. So this is the cochlea are the main functioning for the hearing. Okay, so cochlea is present in the inner ear, and this also you can see the nerve. This is the nerve connected in the cochlea and labyrinth. Okay, so I told the pinna, that is the external ear, and the middle ear and the inner ear. So apart from the external ear, just pinna and ear canal involved in the external ear. Also, you can say that this is the helix and this is the antihelix, this is the lobule and this is the concha. Okay, so in the whenever you study in the detail of the external ear. And now I move on the ototoxic drugs. Okay, so ototoxic drugs means the these drugs used using in the ear disorder purpose, like your eardrum has rupture and like any passage are removed from your ear. Also, you have difficult like tinnitus and you feel the vertigo. Okay, and also infection in the your inner ear. That is the ototoxic drugs. Also, a lot of these drugs also can used in the like loop diuretics and non-steroidal anti-inflammatory drugs, anti-malarial drugs, anti-neoplastic drugs, and miscellaneous. So all these drugs are the ototoxic. Their drugs produce the ototoxic. That means the toxicity in the ear. So our theme is the ototoxicity. So these drugs induce the ototoxicity. And I will discuss detail about the antibiotic, what is the which type of the drug involved in the antibiotics and loop diuretics, and also non-steroidal anti-inflammatory drug and anti-malarial drugs, anti-neoplastic as well miscellaneous drugs. So apart from the aminoglycosides, this is the type of the antibiotic, and streptomycin, kanamycin, and neomycin, sulcamycin, gentamicin. So these all are my group of the mycin drugs involved in the aminoglycosides, and the enter into inner ear by unknown mechanism, secreted into perilimbs by spiral ligament, endolimb, and like stira vascularicis. That means so this aminoglycosides, I mean the mycin groups also produce the ototoxicity for the cochlea. Okay, so just you have to learn which type of the drug, that means the mycin group. Okay, so you can learn, you can you have to know the mycin group like kanamycin, neomycin, and amikamycin. Another cochlear toxicity, like also just this is the clarified amikamycin and kanamycin, and then vestibular toxicity, streptomycin, gentamicin, and sisomycin. Okay, so I mean to say the all mycin groups of the drugs can produce the ototoxicity in the inner ear, and your cochlea has totally damaged. That means the you can permanently loss of the ear. And then you can see the aminoglycosides, that means the mycin group of the medicine. Cochlear toxicity. So what is the cochlear toxicity? So outer hair cells loss first in basal term, then to apex. Inner hair cells loss. So in this picture, some apex like this structure. That means the whenever you take the mycin group of the medicine, that is the aminoglycosides, then your cochlea has toxicity. That means your outer hair cells totally damaged. Okay, so when the detail in the cochlea, So main outer hair cells and the inner ear cells. Two hair cells present in the cochlea. First inner ear and second is the outer hair cells. First is the outer ear cells and second is the inner ear cells. So these main hair cells are functioning the hearing. Okay. So whenever aminoglycoside, that means the myosin group of the medicine you take, then your cochlea has totally damaged. That means your hair cells damaged. Okay. If your hair cells damaged, that means you can your doctors prescribe the hearing aid. Why? Because this is the type of the neurological problem, because nerve cells has totally damaged. So in this case, no, in the medical science, no any type of the drugs has discovered for the recovered to your hearing. Okay, because you lost the hair cells, outer ear cells and the inner ear cells. This is the type of the nerve. Okay. Also loop diuretics like ethacrinic acid. Okay, so this is also type of the loop diuretics. And loop diuretics, that means the drug applied on the heart failure and also chronic kidney disease. Okay, so also this type of the drugs as prescribed by the prescriber. Okay, so that also this can produce the ototoxicity. And then you can see in these slides the usually tinnitus, temporary and reversible as an H. That means the sensory neural hearing loss. Okay, so just I told you, if your prescriber diagnose your ear has sensory neural hearing loss, that means your cochlea functioning has totally damaged. That means that no medication, no surgical process for recover your hearing. Okay. So in the end slides, I will also discuss what is the management and what is the recover of the hearing. Okay, so one, I think you some know that the cochlear implant. Okay, so cochlear implantation can be done, but what is the cochlear implant? Just I will discuss. Okay, so just I am telling that what is the ototoxicity drug and which type of the drugs involved in the ototoxicity. Also salicylates and NSAIDs drugs. Okay, and in this normal histological no hair cell loss, clinically tonal high frequency tinnitus, that means the 7 to 9 kilohertz. Okay, so that means what is the tinnitus? You feel you feel like tinnitus, that means the ringing sensation in the ear. Okay, ringing sensation means कि आप कभी-कभी लगता है कि कान में जो है घंटी सी आवाज आती है, राइट? जैसे लगता है कभी-कभी कान बज रहा है या कान से आवाज आ रही है, that is the tinnitus. ओके, और कभी-कभी सेंसेशन फील होती है, सनसनाहट की आवाज आती है कान से, तो उस तरह का फील जब होती है तो उसको कहते हैं tinnitus. एंड tinnitus इज द आल्सो सिम्टम ऑफ द ओटोटॉक्सिसिटी. दैट मींस कि आपने कोई टॉक्सिक ड्रग्स लिए हैं, कोई मैसिन ड्रग्स लिए हैं, आपके प्रिस्क्राइबर जो आपके डॉक्टर्स हैं, आपको इस तरह के ड्रग आपने लिए हैं, लंबे समय तक आपने वो ड्रग को यूज किए हैं, तो उसका साइड इफेक्ट से क्या हुआ है कि आपको tinnitus हुए हैं. तो दैट मीन tinnitus हुए हैं, तो ये भी सिम्टम है कि आपका इनर ईयर मीन्स है कि आपको फंक्शनिंग वो डैमेज हो रही है, तो आपको वहीं अलर्ट हो जाना है कि tinnitus फील हो रही है, रिंगिंग सेंसेशन फील हो रही है, तो दैट मीन्स हमें वो कोक्लिया कहीं वो फंक्शनिंग में प्रॉब्लम तो नहीं आ रही है, तो इमीडिएट आपको डॉक्टर से दिखाना चाहिए. तो यहाँ पर इस केस में क्या होता है कि इट शो द माइल्ड टू मॉडरेट सेंसरी न्यूरल हियरिंग लॉस. ओके, and also quinine drugs also show that ototoxicity. Okay, and high pitched tinnitus. अब यहाँ देख सकते हैं क्या क्या हो रहा है, क्यूनिन ड्रग जो है वो हाई पिच tinnitus हो रही है, मींस कि बहुत ही हाई पिच. हम जानते हैं कि फिजिक्स में सभी कोई पढ़े हैं 10th 12th में कि साउंड के जो क्वालिटी जो पैरामीटर्स होते हैं, पिच, लाउडनेस एंड क्वालिटी, ये 3 पैरामीटर्स होते हैं, पिच, लाउडनेस एंड क्वालिटी, जो कि हमें उसका डिटेल्स हम पढ़े हैं वहां पर. तो यहाँ पर क्यूनिन ड्रग्स जो है क्या कर रहा है कि वो पिच को नहीं, मीन्स की हाई पिचे जो हो रही है उसको आप शो करिए, मीन्स कि आपका जो टोन है, जो आपका पिच है, जैसे कहते हैं ना कि सबसे ज्यादा हाई पिच जो होती है वो चिल्ड्रन की होती है ना, जैसे चिल्ड्रन हो गए, मेल हो गए, फीमेल हो गए, तो सबसे जो लो पिच होती है मेल की होती है, देन फीमेल की होती है, और सबसे हाईएस्ट जो पिच होती है वो किसकी होती है, चिल्ड्रन की होती है. इसलिए जब कोई बच्चे चिल्लाते हैं बहुत जोर से तीखी और सुनाई देती है कान में, देन फिर वोमेन की होती है पिच. तो दैट मीन्स जो आपका tinnitus होगी वो क्या होगी, हाई पिच होगी. और ऑल्सो ये क्या करेगा, सेंसरी न्यूरल हियरिंग लॉस को शो करेगा. एंड ऑल्सो एंटी न्यूप्लास्टिक एजेंट्स जो भी हैं वो भी इसको शो करेगा. ठीक है, ऑटोटॉक्सिसिटी को। और ऑल्सो कुछ एंटी माइक्रोबियल है जो कि CSOM है, आपका ईयर ड्रम जो है वहां पर अगर रप्चर हो रहे हैं, वहाँ इन्फेक्शन हो रहे हैं, तो उस कंडीशंस में प्रिस्क्राइबर जो है वो CSOM इस तरह से वो लिखते हैं। ओके, so also topical antimicrobial, polymyxin B, chloramphenicol, neomycin, gentamicin, ticoracillin, vazocidin and ciprofloxacin, ये सभी जानते हैं। ciprofloxacin ड्रग भी क्या है, हम mainly used on the for the infection purpose। ओके, so ये सब मैंने आपको दिखाया कि ये सब जो है ड्रग हैं जो कि मेनली टॉक्सिक इफेक्ट को शो करते हैं, और इससे क्या हो सकता है कि हमारा जो हियरिंग है वो टोटली लॉस हो सकता है। और मैंने भी जस्ट बोला कि अगर आपका कोक्लिया का अगर फंक्शन अगर आपका डैमेज हो गया तो अब आपका कान का जो हियरिंग है वो वापस नहीं आ सकता है, क्योंकि आपका न्यूरल आपका वहाँ प्रॉब्लम हुआ है। वहाँ पे जो हेयर सेल्स है, अगर आप बहुत ही ज़्यादा नॉइज है ना, हेडफोन यूज करते हैं अगर हम लोग, तो अगर आप कंटिन्यूसली अगर नॉइज एक्सपोजर होगी तो उस वजह से भी आपका कोक्लिया डैमेज हो सकती है। तो इसलिए हम बहुत ही ज़्यादा नॉइज एक्सपोजर में भी हमें नहीं रहना है ना। ऑल्सो इस स्लाइड्स में आप देख सकते हैं कि नॉइज एक्सपोजर नहीं होना चाहिए। ओके, तो वहाँ उस मेरा कहने का मीन्स है कि जो हमारा अभी जस्ट मैंने फर्स्ट में बोला था कि ईयर के 3 पार्ट्स हैं, एक्सटर्नल ईयर, मिडिल ईयर, देन द इनर ईयर। अगर आपका प्रॉब्लम इनर ईयर में चला गया, आइदर वो आपका इन्फेक्शन हो, आइदर आपका वो मेडिकेशन से टॉक्सिसिटी हुआ हो, अदर आपका कुछ एक्सीडेंटल कुछ हुआ हो, अगर आपका इनर ईयर अगर वहाँ पे कुछ डैमेज हुआ हो तो वहाँ पर आपका जो हियरिंग होगी वो टोटली परमानेंटली लॉस हो जाएगी, और उस कंडीशंस में आपका हियरिंग वापस नहीं हो सकता है। तो जस्ट हम कुछ मैनेजमेंट है जो मैं एंड में भी डिस्कस अभी करने जा रहा हूँ। और ऑल्सो ये क्या है कि क्लेफ्ट लिप एंड पैलेट चाइल्ड है जो कि मेनली एक कंजेनिटल, दिस इज द टाइप ऑफ द कंजेनिटल एनोमलीज, एंड देन ऑल्सो इसमें क्या होता है कि इस चाइल्ड में क्या होगा कि इसको हियरिंग टेस्टिंग भी करना नेसेसरी होती है इस केस में। और ऑल्सो ये क्या हुआ जो क्लेफ्ट लिप है, और ऑल्सो क्या होता है क्लेफ्ट लिप एंड पायलेट, तो इस कंडीशंस में इसको सर्जिकल किया जाता है। अदरवाइज इसे अगर हम छोड़ देंगे तो क्या हो सकता है कि इसके साथ साथ में हम इसका हियरिंग भी कह नहीं सकते हैं कि लॉस है या फंक्शनिंग हो रही है। और अगर हियरिंग फंक्शनिंग हो भी रही है तो क्या होगा कि इसको बोलने में प्रॉब्लम आएगी, है ना। उसको स्पीकिंग में इसका फ्रेश स्पीच नहीं आएगी, जैसे हम लोग नॉर्मल स्पीच करते हैं तो इसका नेजल स्पीकिंग आएगी, लगेगा कि कुछ नाक से बोल रहा है। ओके, तो ये क्या हो गया, कंजेनिटल एनोमलीज हो गया, जिसमें कि मेरा जो जैसे थीम था कि कंजेनिटल डेफ एंड म्यूट। ओके, अगर इस तरह के अगर बेबी चाइल्ड अगर बॉर्न होते हैं तो हमें इमीडिएटली इसका हियरिंग चेकअप भी कराना चाहिए और इसका सर्जिकल भी कराना चाहिए। अगर हम सर्जरी नहीं कराते हैं तो बाद में ऐसे बच्चों को स्पीच जो होगी वो क्लियर नहीं होगी। और ये तो वेस्टिब्यूल का था, ठीक है, जो कि इंटरकनेक्ट था कोक्लिया से, और जो मैंने स्टार्टिंग में बिगनिंग में ही बताया था। और ये आप देख सकते हैं, यू कैन सी द कोक्लियर हेयर सेल्स, दिस इज द मेन इनर हेयर सेल्स, बिगनिंग आई टोल्ड, एंड द आउटर हेयर सेल्स, दिस इज द आउटर एंड द इनर ईयर। तो मेनली damage in the inner ear, I mean in the cochlea, so this inner ear cells and outer ear cells. Okay, and then the what is the management part of the for the hearing loss. Okay, if you have the hearing loss like the and find the like censoring the hearing loss and the permanently total damage, but hearing loss, I mean if the patient, I mean the child age is the not more than 5 years, below the 5 years, that means you can surgical in the surgical process, that means the cochlear implantation. So this picture of the cochlear implantation, so you can see that this is the cochlea. So just I told in the beginning that means the cochlea has totally damaged due to the ototoxicity. So in this case now your ENT surgical process, okay, so ENT surgeons the implant some cochlear implant. So in the implant, that means the some electrodes. Okay, so these electrodes put in the cochlea. Okay, then इसमें क्या होता है कि ये जब इलेक्ट्रोड्स जब इसके अंदर में सर्जिकल प्रोसेस जब इसमें चला गया तो उसके बाद क्या होता है कि इसमें इलेक्ट्रोड्स ये फंक्शनिंग करता है। अब आपका ये डैमेज हो चुका है, इसका फंक्शनिंग खत्म हो चुका है, क्योंकि ये कनेक्ट फिर ब्रेन से होती है। जब हम सुनते हैं तो वो क्या होता है, जो सिग्नल होती है ना वो ब्रेन को जाती है। अब हम कहते हैं कि अगर जो वायर हमारा है, जैसे कहते हैं इलेक्ट्रिसिटी जो वायर है, अगर वायर में जो वो अगर जल जाता है, ठीक है, तो क्या होगा कि इम्पल्स जनरेट नहीं होगा। क्यों नहीं होगा? क्योंकि वहाँ पे वो जो वायर है वो जल चुके हैं, वो डैमेज हो चुके हैं। तो वही इस केस में होता है कि जो हमारा हेयर सेल्स था वो ड्यू टू द ड्रग एक्सपोजर से वो क्या होगा, डैमेज हो चुका है, आइदर ऑल्सो इन्फेक्शन से भी। तो इस केस में ये क्या करता है, जो इलेक्ट्रोड्स है, जो इम्प्लांट है, जैसे हार्ट में इम्प्लांट होती है, आई का भी इम्प्लांट होता है, तो वैसे ही कोक्लिया में भी इम्प्लांट किया जाता है। तो इसमें क्या कर सकते हैं कि इसमें आपके मन में क्वेश्चन हो रहा होगा कि इसमें जो आर्टिफिशियली इलेक्ट्रोड्स को हमने जो फील किया, क्यों ना हम इस कोक्लिया को इम्प्लांट कर दें। तो ये क्या है कि ये इतना सेंसिटिव होता है कि हम कोक्लिया को इम्प्लांट हम नहीं कर सकते हैं, इस ऑर्गन को हम एडॉप्ट नहीं कर सकते हैं। तो इस केस में एक आर्टिफिशियली एक इलेक्ट्रोड होती है और इसके अंदर पुट किया जाता है और इसका रोल खत्म हो जाता है, और इसके अंदर का जो हेयर सेल जो डैमेज होते हैं वो अब ये वर्क करता है, ये जो इलेक्ट्रोड्स है। अब जब आपका बाहर से जो साउंड आएगी वो साउंड को कैच करेगा ये, और उसके बाद ये इलेक्ट्रोड उसको स्टिमुलेट करेगी, देन फिर वो इलेक्ट्रोड इस वायर के होते फिर आपको ब्रेन को जाएगी। बट दिस प्रोसेस को करने के लिए अगर बच्चे का एज अगर बिलो 5 ईयर्स है, 3 ईयर्स है, 2 ईयर्स है, तो उसमें यूजफुल होती है। अगर मैच्योरेशन अगर बढ़ चुका है, माइंड का मैच्योरेशन हो चुका है, तो उस केस में क्या होता है कि थोड़ा डिफिकल्ट आती है, ठीक है। अदरवाइज इसमें एक हियरिंग एड होती है, वो आप उसको यूज कर सकते हैं। तो आप इस तरह के कभी आप कहीं भी आप मार्केट प्लेस में, मॉल में या किसी प्लेस में आप देखे होंगे किसी चाइल्ड में इस तरह के इम्प्लांट लगे हुए होते हैं। ओके, सो ये क्या आपका कोक्लियर इम्प्लांट है, और यहाँ पे देख सकते हैं एक डिवाइस है, इसको सर्जरी किया जाता है, पिन्ना को पूरा काट के निकाला जाता है। ओके, एंड देन इसके अंदर इलेक्ट्रोड, इसके अंदर में क्या इलेक्ट्रोड ऑलरेडी फिट किया हुआ है कोक्लिया में, और इसका जो पिन्ना है इसको सर्जरी करके स्टिच किया जाता है। देन फिर एक अदर डिवाइस होती है, ये डिवाइस यहाँ पे कैच किया जाता है। इसके अंदर में क्या होती है, मैग्नेट होती है, यहाँ पे चिपका हुआ है, और क्या होती है, अब आपका जो ये ईयर है ना, इस ईयर का रोल खत्म हो चुका है, और ये आपका जो पार्ट है हियरिंग एड है, अब आपका कान ये हो गया, अब आपका ईयर ये नहीं है, अब आपका ईयर ये हो गया। जब साउंड इसमें से आती है तो फिर इंटर होकर के इसके थ्रू होते हुए आपको फिर ब्रेन को जाती है। तो ये इसका मैनेजमेंट पार्ट था। और जो दिस इज द रेफरेंस ऑनलाइन पिक्चर गूगल्स एंड ऑल्सो एसेंशियल ऑफ ऑडियोलॉजी girlfriend 4th edition. Okay, thank you. And then if you have any query and question know about the type of the disorder of the hearing, also you can ask me. If you have any doubt or any query, कुछ अगर आपको लग रही हो कि कुछ इसमें से आपको जानने की इच्छा हो रही है, तो यू कैन आस्क मी। बिकॉज मैं कुछ प्रोजेक्ट वर्क किया था, मैं एक गवर्नमेंट हॉस्पिटल में एज ए जूनियर ऑडियोलॉजिस्ट में वर्क कर चुका हूँ, ठीक है। उसमें 3 इयर्स 5 इयर्स में गवर्नमेंट का प्रोजेक्ट था, उस प्रोजेक्ट में मैंने वर्क किया, और बहुत सारे मैंने बेबी पर मैंने एक केस स्टडी किया है, जिसमें कि उसमें हियरिंग मैकेनिज्म और ऑल्सो उसका ड्रग का इंड्यूस्ड किस तरह से हुआ है, तो उसमें मेरा वर्क हुआ है। ओके,
So I think everybody is, feeling that this is not from assignment presentation, but I had a request from Devraj for if he can present his project work. So this is just I allowed to, have a extra session or something new
yes ma'am,
to understand, right? So if we have time and if you are willing to present your interest of topic or maybe your work or a project, you are free to present it whenever we have such sessions, right? So
okay,
it will, others can also be benefit.
Yes, ma'am. Also, I have prepared the presentation about the BIIS, ma'am. So which also I have sent in mail, ma'am. BIIS and also COVID, ma'am.
Okay, no problem. So one group is still Narmada. Hello. Who is left to present the assignment? Which group is yet to present.
There is a message from Norma.